Okay, so it gives me a lot of pleasure to introduce you people from my own alma mater. Okay, Alok sir here is from JNU. Anjali ma'am is here from IIT IGTW. So let me introduce you both the resource persons who are here. Okay. Hello. Hello. So, so let me start with introducing Dr. Anjali ma'am. Okay, Anjali ma'am is presently assistant professor at IGTW at New Delhi in the Department of Computer Science. Silence. Shruti said join online from there. Question you join from online. Okay. So she is working.
This guys. So when you fuse the biomedical sciences and the data science together, it becomes and known as the field of the bioinformatics, right? So today we are just trying to bring before you few of the fundamental concepts of phylogenetics, right? So what is phylogenetics? What are the different methods to build some phylogenetic tree and all, which will be given by Dr. Anjali here. Okay, and I'll be just trying to give some assistance and to bring the session best before you. Okay, and by the end of the session, we'll be more than happy to take your doubts, and it will also be great if you can have your doubts among the session. Right, Anjali, it will be good if they can ask the doubts in between as well, right?
Yes, totally fine.
Right. So please, guys, feel free to have as many doubts as you can in between the session. Okay. Because by the end of the session, we are looking that you should have a little bit idea like what is phylogenetics, and you should have a interest and motivation to understand and go into the field of evolutionary biology, because phylogenetics and all comes under the branch of biology named as the evolutionary biology. Okay, so as Dr. Ajay has already introduced me, like I'm currently running this particular startup, TechMedBuddy, where we try to train the student coming from the life sciences background for biomedical science, biomedical data science, healthcare data science, and a number of different codes or different branches. Okay, and we are trying to collaborate and bring expertise of the people from the academia, from the research, and from the industries as well. Okay, so without wasting any further time, let me hand over the stage to Dr. Anjali, and you can continue then. Thank you so much. Yeah, Anjali, you can continue.
Yeah. Thank you so much, Alok and Dr. Ajay, for this wonderful opportunity. Hello.
Let me
Uh,
mute all of them. Yeah.
I thought there was some question, that's why I stopped.
No, no, it was some mute basically. Yeah.
Okay, okay. Thank you so much, Alok, Dr. Ajay, for this wonderful opportunity to interact with the students. And the topic that we'll be discussing today will be phylogenetics. And before that, though Dr. Ajay have already introduced me, I'm currently working as an assistant professor at IGTTU Delhi, and I did my PhD, postdoc, all these fancy things and degrees, but mainly primarily interested in my background is computer science, but I switched to my research is in bioinformatics, mainly healthcare domain, around genomics and multi-omics studies. So let's dive into the section today. So, Alok, if you can please unmute Dr. Ajay's screen so that if
Okay, okay. Just a minute. Sorry.
yeah, if I have to ask something, then I can ask them.
Yes.
So, how many of you are really aware about what phylogenetics is?
I'm just asking you something. Please respond.
Because I have been told that you know the basic definition of what phylogenetics is, basically, right? You can please raise your hands. How many of you are aware about phylogenetics?
They are aware and they are aware of the basics.
Okay. Uh, even that, I'll just tell you what, in a simple way, what phylogenetics mainly is. Imagine you have a giant family reunion, and you have to figure out how everyone is related. There are your ancestors at the top, branches, connections, leaves, basically the present scenario. Your family may have some pets, right? You yourself, that is the current scenario. So basically, it's being like a detective of your history of life, where rather than having those old photos and everything, where you try to figure out who's connected with whom, you use DNA, body structures, fossils, right? So simply, phylogenetics is that, and basically understanding its science of ultimately finding the tree of life, where who is connected to whom. And let me quickly share my screen. I'll walk you through a small presentation. Uh, my screen is visible, sir?
Yeah.
Visible.
Okay. I may not be able to see you all. Uh, I don't know how to do picture in picture here, Alok. If you have some idea, then
No, like, no, when you are presenting, you cannot.
okay, here I can't. Okay. Uh, so if there are any queries or anything, you please let me know if somebody.
Sure, sure, I will.
Okay. So, phylogenetics, this discussion today will be going through how organisms are related through their DNA, some hands on practice sessions, how to code via Python. Python or anything wherever you are comfortable, you can use that. And definitely, if you don't have any prior experience in terms of what phylogenetics is, then that's also totally fine. We have designed today's session to be very generic, and anyone who's not having good understanding or have some prior experience in phylogenetics is also be able to understand. So what essentially is phylogenetics? As we discussed, it's like drawing ultimate family tree of life. It's not just for humans but for animals species, how over time we also evolved. So study of evolutionary relationships basically. And where it is used, why is it important? Classification first. Basically, use DNA sequences for accurate species classification. For example, if I would like to understand, that's why we say that we have been evolved from apes, because we track over the period of time how our DNA, how closely it matched to certain species. So it helps us to elucidate those things. Now, forensics, crime scenes. Let's say if somebody fall ill, so what bacteria, what viruses have been injected or infected to that person, I can track their genome, right? To see how it have been impacted. Different people may react to different species or viruses differently, right? COVID have shown us that varying degree of responses based on these are personalized behaviors, right? Disease tracking as well. So how we knew that it came from bats? It did not impact them. So pathogens, what is there? Origin levels, traces, how it outbreak. It really helps inform public health policies and decision making. Conservation, basically, let's say if I have to see how much two species are related. One day, if I have seen two tiger or cat species, let's say tigers, lions, or leopards, how closely they are related, that will also help me to design certain conservation policies. Let's say if these are very similar, can some similar conservation policies work for this group of species or not? Am I clear till now? I hope I'm audible and understandable enough.
Yes, ma'am.
Okay, thank you. So, and then the bioinformatics, it's basically when you are understanding something, its data is so huge. We know faster files or DNA, genomic data in itself, it's of GBs and TBs when we have to look for a certain population or people, right? So, you need stronger algorithms. Just mere by manual calculations, it's highly and it's a really impossible task to analyze those data. So, bioinformatic approaches where you design algorithms process to generate insights out of that data. So that is also one part of our discussion here. So basically, where phylogenetics is important: classification, forensics, disease tracking, conservation, and then bioinformatics and AI development specific for evolutionary sciences. Now, brief continuation of that, what we have discussed right now. See this picture over here. You see, 13 million years ago, all of these used to share a lot of DNA, right? There were hardly any differences in terms of, see, minor differences, less than 1% or around 1%. That's their diversity in humans as well. That much, that's why you take 1,000, 10,000 people data to make that normal reference, right? Some mutations are naturally occurring. For diversity reason, it should be there, right? So around 13 million years ago, you see this knob here. So these were the common ancestors. Then around 8 million years ago, you see these branches. This says where certain lineage of species diverged from the other. Like orangutan, say, if you see, I hope everyone is okay with a mixture of Hindi and English, or shall I be using only English?
Anything is fine.
Okay,
And
fine.
they speak only English. And they are
Uh,
not diverse with Hindi. So only English will be better.
okay, I'll take care of that. So let's see, when you see it 8 million years ago, that this four species, including humans, gorilla, chimp, bonobo, these were separated from orangutan, right? Then around 6 million years ago, even gorillas say these three species have been diverged, like chimps, bonobos, and humans. So this is how it helps you to track around the lineages, that how much their DNA have been changed, and based on the similarity, and we'll uh, study in our subsequent lecture today what are the methods to generate these trees and what principle they work, etc., etc. Okay, so a quick recap, how and why is it important from this figure as well: evolutionary insights, disease tracking, what are the COVID-19 mutations and how it is impacting humans, because bats, it's still there but it's not impacting them, it's not making them ill, right? So there is something which is being done differently at human level. Then forensic science, and of course genetic engineering as well. Now, some basics. When we are building these sort of trees, roots, to understand the better relationship, so why only these trees, branches? Because this sort of data structuring gives me a better understanding for this, right? Uh, when you arrange your stuff at home, for example, you may use different sections. Cups, you may use certain different things. Classes, you may use certain different things. Similarly, in biology or any other domain in computer science or bioinfo, different type of data, different type of problem need different type of structures to represent that data, or to effectively make sense out of that data. For evolutionary things, tree make more sense rather than just simple sequential things, like, right, like simple sequential boxes, which in computer science terms we say arrays boxes, right? So this is a more structured way here. That's why trees. So in phylogenetics, it's mainly two type of trees: rooted versus unrooted. So rooted is something which you can see here, or which we have seen earlier as well, this figure, where you have a common ancestor and then you have a direction of evolution as well. Okay, this is at top and you are driving something down there, right? It helps you to trace back to the evolutionary origins basically. Now, unrooted trees are, these lack some common ancestors, but they do show some relationship between this species, not ancestry per se. So, main key difference is rooted. This explains evolutionary history, while unrooted, they mainly focus on genetic relationships. Some real life example, if I say, let's say if I have to track how humans have evolved. then rooted will make more sense, because I have to track that species ki first ancestors was who, and over the years across the branches how everything across the species was evolved. Unrooted basically where you have to do comparative genomics. For example, I don't have common ancestors, but I have to see how two species are related. Let's say one day, if I have to compare the genomes of bacteria to see how closely one is related to another. If I get some new species of bacteria, I'll compare it with every species to see how closely it matches to certain species, right? So that these are the two applications for rooted and unrooted. Clearly having some restrictions where one will make more sense as compared to another. Now, some common terminologies that might make sense for you for tree anatomy. Like for our anatomy, we have hands, eyes, everything. For trees, you have branches, which are your evolutionary pathways. Pathways is over the time how things are changing. Nodes, these are the nodes. Here you can see ABCD. Like here, these are the nodes. Then, which have common ancestors, and then leaves, current species. Here, these are ABCD. Here, these are these humans, current species, right? So this is some basics about what phylogenetic tree is. Now, so till now we understood ki what tree is, what is rooted versus unrooted, right? Basic terms. Now, how we essentially make these trees? Are there any methods? Of course, there are some methods for building phylogenetic trees. Abhi, there can be different ways. If I have to make a tree, I can pick, like while you are arranging your clothes, one would like to see, I'll segregate them with colors. One would see, okay, I'll put my winter clothes here, others here. One simple example. Or let's say I would like to keep my t-shirts here or my trousers there. So there are different ways how you can arrange and build something. Similarly, here there are different methods that we'll be discussing today for building phylogenetics tree. One is neighbor joining, NJ. Quick distance based method है, where iteratively you join the closest species based on genetic distance. What do I mean by genetic distance? That comparative genomics, right? How closely, how similar they are. For example, uh, where you have unequal evolution rates. For example, if uh, I understand that it's basically work on a notion that every species may not evolve with an equal rate, right? Some may evolve faster, some may evolve quite slower as compared to other species. There you use neighbor joining. It's flexible and work with it finds the closest species and join them first. And when to use it? When species evolve at different rates. And UPGMA is unweighted pair group method with arithmetic mean. What it is, basically it assumes that mutation rates or evolution rates, these are almost evolution rate is equal. That is this assumption which UPGMA method makes. It's quite fast and simple. It's basically group species with similar DNAs first, with assuming equal rate of evolution, or we have some indication of whether species evolve at a similar rate or not. Then you use it to build those trees. Now, maximum parsimony is to create the trees that need fewest changes. Like you look at you pick those species together which have least DNA changes over time. Try to minimize the number of changes in DNA to build the tree. Like the closest one will have minimum mutations across their DNAs, right? Like when you want the simplest tree, which is fewest changes across the branches. Maximum likelihood is, it's basically where you it's the most accurate and most reliable form of making a tree based on evolutionary rules. Basically, it's a purely math based method. So it's test many possibilities and pick the one which best fit your data. Your data can be anything, right? Evolutionary files, years, how the mutations are different, everything. When you need the most accurate tree, when then you use the maximum likelihood. Basically, when I'm saying ki certain rules they use, what rules it can be? Substitution models, rate of change, and the tree topology. Substitution models are how genetic material is changing over time. I may want to look at it like nucleotides, amino acids, how they are changing over time. Rates of change, how quickly or slowly these different sites are changing. See, they are two different things. One is dealing with genetic material changes over time. One is how quickly and slowly it is changing. Rates of change have that time component as well. Tree topology is the structure and branching pattern, how it will be. Is it what I'm going to follow? Uh, this will be out of scope of for the today's lecture, but we can definitely take up some another class where if you would be interested in some more understanding around these methods for sure. But this is how a basic understanding what are the different famous methods. There are other one or two methods, but these are the mainly used methods for building phylogenetic trees, and some basics about how they are used, when they are used. Fine. Now, okay, I think this slide by mistake I have put it. Let me remove this. Yeah, again, we did discuss about a lot of things, right? But then, uh, I'm building some files, trees, that's fine. But I also need some data, right? Your data can be in your FASTA sequences. This can be in any other Newick sequences. So, what data are permissible or using these data, how you can create these trees, these are some of the formats. Then again, quick review: Newick, Nexus, NexML, PhyloXML, CDAO. These are some of the phylogenetic tree formats. Basically, Newick is a text-based format. You see this example. AB are basically, like say here, if I have to, AB is your CNB here. CNB, because these are grouped together at a certain level. CNB and here C is something like human. So A or B are very closely related because they are in enclosed in one bracket. C is more distantly related to as compared to A and B. So this is Newick format. Nexus is, it's like sequences mainly around a file may contain data as a tree structure and also may as a DNA sequences, FASTA sequences or any other. NextML, these are purely XML based format. You look at this example over here. This is how it is represented. Okay, phylum mein A is here, B is here, and then these are showing species A and B. NXML and PhyloXML, these are mainly machine readable. So when we are dealing with computers, essentially everything have to be transformed to 0 1 binary. हमारे लिए for us it is alphabets A B C D numbers 1 2 3. But for computers it's A is also something in form of 0 1s when they understand. So these in a very overview or summary sense, XML and Philo XML, both of these are very machine friendly. So this base formats, these only difference between NXML and PhyloXML, they also have some sort of metadata like branch length. Branch length specifies ki how closely and distantly over the how much time it took for them to uh, dissociate. Here, if you see, right, this branch length, these lengths, right, how distant there was some separation, right? Again, all of these are XML based formats, XML, Philo XML, CDAO. They have again, CDAO have other traits as well, like color. You can assign certain colors also for the traits, for the nodes. So these are some of the formats. Now, if I'll have to start, I have put some snapshots here, but we'll be also going through a Python notebook. I'll briefly talk about what is it and how you can build your own trees, use the data files, right? So our session today will be using Python, but you can use Python and R anything. So this is how the overall console will look like. Console is like your window. You can see here you are typing some codes. This is how your tree will gonna look like. These are some files. So Python actually helps you to automate this tree construction and visualization here. Python is again a medium, a language. Like I'm communicating with you in English, Python is giving you that upper layer abstraction where it is understandable to me also. Like if I'm writing here from Python intro to Phylogenetics, and it is converting at the back end. This has already been coded to make it understand to computers also. Okay, oh, what I meant. It's like a translator basically. Fine. Then there are libraries. What is these libraries are? BioPython, DendroPy. These have already methods. For example, uh, if I'm saying create a tree, there is a command read data. So what read data will gonna tell computer, so that computer can understand what I have to do when somebody is saying read data. All of that information is stored in these libraries, where I just have to write. Some scientists, the people who have built these libraries, they have worked on it. Bioinformatics AI level people, right? We have to use these libraries. They have make this task easier. Ki, okay, this word means this, so computer will understand. Otherwise it's tough to make them understand because they understand a different language from us. Fine. So these are widely used two libraries which have all this information, which I also am able to understand, and they can make computer understand as well what we want out of it. BioPython and DendroPy. Sorry. So how you will install it? This is one code snippet. So I'll quickly just for 1 second I'll switch the screen. So you know this is CO Google Colab. We generally use it. It's like I'm using slides. This is also Google Slides, right? Google extension which provides you to make presentations. Google Colab is a programming environment which is linked to my drive. I have all that data on my drive. These files will automatically saved there. This is a Python terminal where you can write codes, you can run it. For example, if I have to run it, I can run this command. Right, it's running. See, you can see connecting to Python 3 backend engine. That is working on some server where already Python is installed and everything. So this is a Google Colab. It is available for Python, R, C, C++, anything that you will mention. See, it's running. So, what flexibility it gives you, you can share it with your friends. You collectively can work on this. It have already pre-installed things it handles, and currently it also gives you support. Let's say if there is some error, let's say I forget n likhna and I run it. It is showing some error. It will definitely. Right? It's showing error. What I can do is I can open Gemini. What is Gemini? Your AI bot. This is some also flexibility which have been given. I can write it here. It will help you to debug your code. You don't have to Google it all the time. See, it will tell you. You have to change your. There is an error in Python. So it's a fantastic thing if you are working with your codes. Understood? So this is how you're gonna install BioPython. I have put that snippet over here. Once you install it, how you build the phylogenetic tree. Here we are using Nuvik tree format. You see, gibbons are closer to orangutan, right? Then gorilla, bonobo, chimp, and humans are distant. Humans very close to chimp, right? You can tell the branch differences 0, 6, 6, right? Everything. These are the branch lengths. How? So if I see orangutan and gibbon, humans are very distantly related to them, closer to chimp, right? So first you load the sample, you align the sequences, and then you generate the tree. How? Here I have preloaded file. Okay, it's too much of time since I have used it. So I have this cytochrome b.primates file on my here faster file. I'm using it on Google Colab. Sorry, it will gonna be load. What we are discussing here is first you have to load the DNA sequences. It can be in faster file, or if you have Newick format file, you can directly use that file also. Then you have to align the sequences. Once you have the DNA, I think this basic process everyone is aware about. For faster file or DNA sequences, you first have to align them to have some interpretation, right? Correct? Am I audible?
Yes, ma'am.
Okay, thank you so much. Then you generate that tree. Here we are using NJ method. Once we have gone through this, I'll switch to that Python file as well, just to give you a brief how it is done. Now, how you interpret the phylogenetic tree? We have understood it here. Pehle bhi. Branches, what does this mean? Branching patterns indicate how the species are related, be it this or be it this, anything, right? Then the closer two species are in branching, the most recent their common ancestor is. Like here. So if I see these chimp, these are closer to humans than any other species. Fine. Longer branches, greater evolutionary divergence. Like if you see here as well, longer branches, greater evolutionary divergence. Many years ago, you have been diverged from orangutan, right? Then again, humans and chimps, very recent common ancestry, making them more closely related. Gorilla is certain distant, orangutan more, and then gibbon way further than that. So this helps us to have a very see. If I'll just talk about it, if I see at this file format, or forget about this, if it is just sequences, faster sequences, it's very difficult for me to infer something. Whereas if I look at this tree or this tree, I can infer in seconds that okay, these are related. These are that closer. These are how many million years ago they diverged, right? Now, so we understood key Google Colab, Python code, everything you do. But there are tools as well where you upload your faster file online web servers. One is this ITOL interactive platform where see this is the platform. This is how it looks. Where you can annotate, upload your tree, etc. It will give you flexibility, be it any file format you can give, right? Regular plain text, fast as well. And this is how it will gonna generate. You can also customize this control panel. We can do colorings, we can increase the font size. So it gives you that flexibility also. If you want to, Google Colab is one thing. Another thing is, Acha, I forgot one thing. Let's say if you have created this uh, tree in Google Colab, this command will help you to download that tree. Where from Colab you are downloading that Newick file. And I have used the same Newick file on the ITOL web server to build this tree. And it gives you good options in terms of, let's say, if I have to choose the file. I had this Newick file. I am submitting to it. I am uploading it. And it will make this tree. You see these panels, branches, right? I go to advanced sections. I would like to see the branch lengths. It will show you the branch lengths. Then I would like to see something else. Nodes, leaf symbols. It's showing leaf symbols, right? Other functions can be there. Internal node symbols, where every nodes are there, where it's separating, right? You can see the scale as well. You can see how much is the branch length, information, everything, correct? All of these. And there was also one, let's say if you want to sort, it can be sorted, right? All of these are options. If you want to invert the tree, you want to slant everything, right? These sort of trees. So this is a fantastic option as well. Fine. Now, bootstrapping will not go much into the details. It's basically work on the randomly when you try to build trees or multiple times. Is it following the same set of same structure? Everything is, let's say this tree, when I'm talking about gibbon, is always coming out to be 14, 12, 9, 6. This is the same way how tree is generated. These basically assign confidence scores to the branches. uh, it's a good way to have more reliability and in terms of whether trees are consistent or not. So this is one way. We'll not be covering that in today's lecture because of time constraints also, but definitely if somebody is interested, we can plan ahead a detailed workshop on all of these algorithms, methods, what are the details, how underlying functions are working, if anybody shows interest in that. So these are some takeaways. Before closing today's section, I'll go through the Python notebooks as well to take you through how one tree is designed. Right? Just quick summary. Basically, phylogenetics help us to understand evolution and genetics, make tree building easy and fun. Python की मदद से we can do it. Then phylogenetics tree, four methods we have talked about it: UPGMA, neighbor joining, maximum parsimony, fine, and maximum likelihood. Just again, quick iterations. UPGMA, constant rate of evolution they assume. Then ultrametric trees, like all tips equidistant from the root. That is what they produce. Then good for closely related species. That's how where you'll use UPGMA. Neighbor joining, distance based it is, but it does not assume both of UPGMA and neighbor joining are distance based methods. But they do not assume a constant rate of evolution. They can create unrooted trees, better for datasets where evolutionary rates vary. Then maximum parsimony, where fewer evolutionary changes are there, good for very small datasets, and because evolutionary events they club based on where fewer changes are there, right? First. So where evolutionary events are rare, there it is used mainly. Maximum likelihood, we did tell it, based on certain rules, very robust, very accurate, certain statistical models have been used. Now, consider branch length and mutation rates both. So it's computationally a bit intensive, but it's very accurate. So you need to see the trade-off. Now, let's go through this one by one. My screen is still visible.
Yes.
Okay. So we have installed it here. This is how Google Colab. What you have to search as well, na? Just Google Colab. Let's say Python search किया था, RB you can also. So it will open. कि Google Colab में can work in R, you can work in Python. This is a great way. Those who struggle to download IDs or they need flexibility to share their code, work concurrently. So I can put the email IDs here and I can share it with anybody who would I like to collaborate with me and work on the same file. Fine. So here we have installed BioPython and I have given a Nuvic file on myself. I have explained it. Let's say here. And I have given the distances in terms of million years. I have assigning these lengths to branches using Clade, fine, dictionary, and then I am drawing this tree. And then I am saving this tree through this output tree expanded file, which I have already downloaded. I have shown you. Fine. Let's say we run it. You just go here and run it. See, this tree will be generated. It's a very simple task. And if I have to download it, I can download that to my system. See, it's downloaded. It's showing one because I have already downloaded it. When I was coding for it. Fine. Now, this bootstrapping, because this was dummy data for me, here it's showing 0, but a good confidence score would be 0.5 greater than that, closer to 1. That means that much of high probability and confidence score. This is dummy data, that's why it's showing 0 and something. Right? As I've told you, it's based on some random sampling. It's just how much random choices, terminals, right? How much resampling you need to do, branching, all of these things. Details we are not covering here. We'll cover in our one of the next sessions if we plan to go ahead with here. Another example using faster files. Since here I already have this Nuvik file format created, but here I'm doing it from the faster files. These are some IPython I have to install similar to that previous things, right? If you run it one by one. Now if I load my faster file, I have shown you I have already placed on my drive, right? Now if I have to run it, I have given the path. I am parsing it, right? This is loaded here. This is a faster file, correct? Cytochrome P gene, these are the species. Now if I have to align these. So one thing which is important here to generate this phylogenetics is length has to be same. The sequence length. But it is not same, so they have trimmed it to make the same length. So this code is for this trim. What is the minimum common length? And everything is trimmed according to that. Fine. This is some extra. If you already have the similar length, then that's fine. FASTA sequences. So if you run it, see FASTA file sequences.phy name, it has been created, which is phy file. Now then alignment, you remember we discussed it over here. Where was it? Here. You need to first load the sample, then you need to align the sequences, and then you need to generate the tree. So we are aligning it here. It has been aligned, right? Means 9 rows, 290 columns. 290 is the length in that case. Now we understood all of these are distance based. Distance, this is similarity. How much similar or how much differences are there in your FASTA sequence? Basically, location wise, where is it changing, how much similar, dissimilar these are. So we need to create this distance matrix. Identity is one such method. So this distance matrix has been created. Now what we use is using this distance matrix, this constructor.upgma means upgma tree will be built. These biophilo tree construction, all of these have that important information. We don't have to worry about it. Some programmer coder have done it already. These are available. What they'll do is using upgma, they'll create that tree, and down there using nj. So if you create it, you will see here, see, this is based on upgma. Sorry, this is based on upgma, and this is based on nj. What is the main difference? As we have already discussed, assume a constant rate of evolution UPGMA, and neighbor joining does not assume it. Produces altametric tree, lagbhag equal length, right? You can see here, right? Equal length trees. Here it's not the case. More flexible. So this is one such example. Similar for maximum parsimony we have created. Fine. Uh, this is what I had to. I was given till 11:30. I had to deliver. I made accordingly. So I think this is mainly today what I had to discuss. And I will be sharing this material, basically
Great.
notebooks, practice problems with Dr. Ajay itself. So you can share that those practice files or whatever notes we're gonna provide with the students to practice it out further. Use these things in their classrooms. So I think I'm be closing today's session and open to any queries.
Fine. So Anjali, I would like to add here something if you're fine.
Sure, sure, definitely.
Great.
You
Sir, am
please.
I audible? Hello. Hi, sir, am I audible to all? Sir, can you please unmute yourself?
I hope I have used majorly English only. Just
Yeah, right, right, right.
right?
Yeah.
Yeah, because there was some
Yeah,
time.
sir, am I audible now?
Yeah,
Sir,
Rakesh,
actually, sir, your voice is coming very feeble.
am I audible now, sir?
Yeah, yeah, sir. Now you're audible. Great. Okay, fine. So, hello, hi guys. So, can I ask now here that how many of you are very comfortable in Python here? Please raise your hand. How many of you are comfortable in Python?
How many of you are comfortable with Python? I think you had your Python programming in second year, right? So they had one Python programming course, so they do know a little bit about installing and how they do have beginner level Python skills.
Right, right, fine. So, guys, see here, like as Anjali, Dr. Anjali has already covered all these fundamental things. Now you really need to think and ask few of the fundamental questions, right? Like due to the time constraint, we were not allowed to basically go into very deeper, but you need to ask few questions. Like for example, when we are creating the distance matrix, so like we said here, identity is one method. Then PAM matrix is another method. Then Blossom is another method, right? So see, like, doesn't matter like which method are you going to use. What matters basically is that how the method is working behind the scene. Okay? Because we are in the learning phase, and being in the learning phase, you really need to know that how this particular matrix is being calculated, how this matrix was created, what was the need for creating this particular matrix, right? Because if you are already having Blossom 62 matrix, then what was the need for generating another matrix, PAM 250 matrix or PAM one matrix, right? Because these are the real questions that being a evolutionary biologist you need to ask. Okay, guys. Now let me give you one question. Suppose that now, if you have to design a complete workflow, if you have to design a complete workflow that you are given some 10 sequences, you are given some 10 sequences, and these 10 sequences are for a particular specific gene. Okay. And now suppose that the gene of the interest here is BRCA1 or TP53 or FOXA1 gene, right? And now I'm going to ask you a question that you need to build a phylogenetic tree. You need to build a phylogenetic tree using these 10 sequences. Okay. And you have to basically make use of different matrices, distance matrices like Blossom 62 and then PAM 250 or some other matrix basically. And then you need to visualize your trees and then look at the changes, like what is the change that we are obtaining, okay, and how one tree is different from the another tree. Okay. So these are the kind of the question that you really need to ask and understand that what benefit one method is going to give you over another method, right? Because you are going to just start your career in the like right now all of you are undergrad students, right? So you will be understanding all these things into high detail. And trust me on one thing basically, that whenever you are trying to learn these fundamental concepts, never ever just go with the theory part. Whenever you are going with the theory part, please try to make use of coding. Nowadays, like both R and Python is very easy to learn language. Trust me on this. Okay, you can go to Chat GPT, you can go to any open AI tool, any AI based tool basically, and just write down your query. Like, what do you want to do? And you will be able to get a complete code for that. Okay, so hope sir, like, do I have 5 minutes so that I could show a small demo for 5 minutes to the students? Okay, great.
I think you should go ahead with it.
Yeah, sure. So
We
let me
have
share my
time.
screen, right?
I
Yeah,
don't
yeah.
think anyone is asking some queries. We
Right.
No one raised hand.
After that, we will ask questions to them basically.
Then you should, I think.
Yeah.
We should start asking questions in that case.
Anjali, is the screen visible?
Yeah, it is visible. Yeah.
Great. So see guys, so I've just interested here to motivate you guys how to make use of AI based things into your learning process. Okay. So for that reason, because let me tell you, like I'm not also evolutionary biologist. Okay. So I also studied all these things at my undergrad only. Okay. After that, I have not been making use of all these concepts and all. And I guess Dr. Anjali is also not making use of all these things currently. Right.
Currently not in evolutionary biology, and I think one small point to add here, which Alok is also trying to convey, is that whatever you would like to learn, you can learn with so much of resources available right now. Like while I did my
Right.
PhD, or the people who did it before me, the number of resources that are available is vast at present. Only thing is
Absolutely.
your zeal to learn that thing. Everything
Absolutely.
that we have discussed today, we are not per se totally evolutionary biologists, even your professors, right? So everyone have to keep learning till they are in a particular domain. Even if you go to industry, you go to certain lab, you go to teaching, learning is a lifelong process. So better to start enjoying it and trying your hands on it wherever you can.
Right. Great. So, guys, let me design you something like see just the assignment that I told you basically that we can how we can solve this particular kind of the question, right? So, see, like I've this is also known as the prompt engineering, basically, guys, that I'm trying to show you and just motivate you like how you can make use of all these AI-based tools to enhance your learning process. Just to enhance your learning process, do not get 100% dependent on these AI tools. Okay, so like see here, you can tell here that you are a bioinformatician. Okay. So first I'm going and instructing this particular AI that you are a bioinformatician. So now this particular tool knows that or it will try to fetch all the information which are required for a bioinformation basically. Okay. So this line is for that particular purpose and instructor as well. Instructor as well, right? Now, you need to understand the concept of, suppose that UPGMA method. Suppose you want to understand the concept of UPGMA method, and then you know you want to generate a tree using that UPGMA method and visualize that for 10 different, for one particular gene sequence, but for 10 different species, right? So how you can do this particular assignment? Okay. So I've told it that you are a bioinformatician and instructor as well. Uh, you need to explain the concept of UPGMA method using Python. For this, you are required to have a gene sequence of gene BRCA1 and of 10 different species, right? Then you are required to perform MSA multiple sequence alignment and build the phylogenetic tree. Everything need to be done in Python Colab only. So write down the code accordingly. Okay. So now you can see here that I've just instructed it with few of the instructions. Okay. And now it will try to give me something. Now you can see here that it is writing down a particular code for me. Okay. So it is writing down the particular code for me. And now I will just copy paste this code. It might give errors because it is not definitely free from errors. Okay. So now you should go to your particular Colab platform. As Anjali already explained you, like what is a Colab. So Colab is a uh, data science based platform launched by Google, maintained by Google, basically, which makes your life easy for performing all the data science tasks over particular servers. Okay. So here I can go and open a particular Colab file. And then what I'll do basically is that I'll just copy paste the code that I've generated with the help of this particular AI into my Colab platform. Okay. And now you can see here that when I will run this code, I will definitely get some error. So, okay, but you can definitely come across those errors easily. Okay. But that is not my task here to explain the error and all. What I'm interested here basically is that to let you know how you can make use of these AI based tools to enhance your learning, but do not get dependent upon these things. So, see here, it has generated the complete code for me within minutes, right? That is not a big deal. But being a student, you are required to know each and every line of this code by yourself. You cannot go to some interview and tell that like I have ChatGPT, so why do I need to know the coding? Okay, because it will give you errors. It will give you errors, and in order to fix those errors, you need to know coding. Okay, so first of all, the task is that you should be able to have a very good domain understanding of the subject, which I had, because of which I generated the assignment. Then the second thing is that because you want to do all these things with the help of coding, so you really need to build good foundations in programming. And being biotech or bioinformatics background students, you need to understand either R or Python. Any one of the languages works. Okay. If you really want to make a career as a bioinformatician, I will highly recommend you to know both the languages, R and Python. Okay. If you want to go for a career in the field of data science, feel free to learn Python. If you want to just be a bioinformatician on like and do only biological analysis, like learn R at a very good level. Okay, guys. And statistical data analysis as well. Like R is a wonderful language which makes your life easy for doing a statistical analysis. Okay. So now you can see here that it generated all the code for me, and it is giving everything like cluster omega command line, distance calculator, distance tree constructor. Okay. So like distance calculator is a function where you can pass the different metric, identity metric, blossom 62, PAM 250, and it will generate the metrics accordingly for you. And then here we are designing the functions. Okay, we are designing the functions here because running functions is always easy. Okay, and now you can see here that I'm generating a list, and then I'm telling here that I'm interested to download the nucleotide sequences for the gene of BRCA1. Okay, and then it is generating, downloading all those sequences for me. And
Look,
then I'm, yeah,
just one small thing. Once
yeah,
you're done with explaining this, I think you
mhm,
should also emphasize on one part where you can use these AI platforms even to understand where your error is and
yes,
what this
absolutely,
code is about.
absolutely,
Let's say
yes,
this code, what Alok is explaining right now, you can paste that in that console and just write one prompt. Please explain this line by line what is happening over here. It will be done. So better to use these resources not to complete your work, but to have a good understanding of where are the problems. It's
right,
your helper guide basically.
yes.
Use it
It's
in
your
a
assistant. It's
sense.
your assistant.
Yeah, it's that's why it's merely as an assistant only. So it's one way, and you should not be even blindly kind of accepting whatever they say, because it's primarily driven by what prompt you are going to give to them. So
Right.
it's not always going to be true. So use your own sense of judgment also.
Right. For example, like here it is already given the explanation basically, guys. Right? You can see here that download the breaker sequence, multiple sequence alignment, compute distance matrix, build UP geometry, visualize the tree. Right. Like as Anjali had recommended to give here, explain each and every line of code by commenting there, commenting in front of them. See, now what it will do basically is that it will comment each and every line. Okay. Now see here basically, guys. Install required packages in Google Colab. Oh, sorry, my just a minute. Oh, my laptop got hanged. I'm really sorry. Is only the black screen visible?
Yeah, it's only the black thing. I
Oh,
think it's hanged.
yeah, yeah. Just a minute. Mm, Chrome disappointed. Just a minute. My laptop got hanged. Sorry. It will not work, I guess. Yeah, fine. So we were here, right? Hope the screen visible now, right, Anjali? Hello.
Yes, this is visible now.
Great. So see here, guys, like I had told it the prompt basically that I want to be commented each and every line of the code as well, right? So now you can see here that it has commented each of the line. Now you can easily understand that what a particular line is doing. And you can use this to like enhance your learning process and learn the things very fastly. Okay. But when it is writing down the codes, also I will highly recommend basically what you should do basically is that take this particular function, try to learn how to execute this function alone. Okay, what is going to be the argument of this function, how this function will return you the matrix or whatever is going to the output of this particular function, and how you should like what is going to the like each step by step process. Okay, because trust me, when we learn the things very in a very great manner when we understand the things one by one in a sequential manner. If you are being taught on like if you are going to study with the help of just your ppts that your instructors or faculties are making use of, trust me, you will not be able to get very good understanding of the things, because see like faculties instructors have their time constraints. They can only make use of they have to teach good number of concepts in a very brief amount of time. Okay, but you have a great amount of time to understand and explore each of the topic in very depth. Okay, so their AI is making a great application, and feel free to make use of these concepts to enhance your learning process at a good level, basically, guys. Okay, so this is what it is here. And now let me show you one thing. With the help of this only, I had created a complete workflow for you guys. It will take only 1 more minute, basically, guys, to explain me. And after that, we will have the Q&A session. Feel free to ask as many questions as you want and any question, basically. Okay. Just let me show you 1 minute. Okay, so this is an assignment that I've created basically. Okay, fine. Genetics workshop. Great. So guys, you can see here that this particular code file I myself generated with the help of ChatGPT. I've not written the code by myself. Okay, it is not possible even to write such long codes in few amount of time. Okay, so now I'll just do what basically if you, like I will go to runtime and I will do run all. And what you will find basically is that it will do everything for me. But let me tell you one thing, to write this code and get all the errors free, to make the code error free, it took me approx half an hour. Because it was again and again giving errors and I had to like repeat the process iteratively for longer number of time to make use of the code, to make the code error free basically. Okay. So now what you will basically see is that it will return you a particular tree, the same assignment that I was telling you, like what you need to do basically. So it is installing all the different packages for now. So pip install minus q conda install and all basically, right? So now it is installed and now it is executing this process. So let me show you see here. So I mentioned here the name of the 10 different species and this is the accession ID. This is named the accession IDs, guys, corresponding to the gene sequence. right? And this is done for a particular purpose. I will tell you. Now here you can see that we are installing muscle. Muscle is a tool for performing multiple sequence alignment. And here we are aligning those sequences. And then we are interested to create the tree basically. So, okay, so you can see here that we are making the distance calculator identity. Okay, and it is doing that particular task. So identity matrix is going to be used. I can change it for PAM 250. I can change it for Blossom 62 and others, and I will get different tree. I might get. I'm not saying that I will get. Okay, and now you see that it has done the task, and now you can see that it has given you a tree where you can see that the different species based on the gene sequence have got clustered together. The species has got clustered together based upon the gene sequences, right? So gene sequences were given. We performed MSA. We got the distance matrix, and based upon that, we performed clustering and got this particular tree, guys. Okay. So this is all about basically like how you can make use of all these concepts and understand the concepts in a very deeper manner and make use of your different AI tools to enhance your learning, but always focus on the fundamentals. Okay, never ever trust on all these AI tools blindly. Okay, because they will give you good number of bugs, errors also so many times, and you have to make use of your learning and understanding basically. Okay, so now we are done on our end. I've created a small feedback form also, but that I will share just once we have the Q&A session over. So over to you guys. We can have a 5 minute Q&A session if you have any, and after that I can share the feedback form. Just a minute. Two meeting. Great.
Anybody?
So fine. So we are done, sir. Like now. Yeah.
Thank you, sir.
Thanks.
Any queries? So basically, ma'am has just told what is the importance of using the phylogenetic trees and what applications
Alok,
they
you
have.
can stop recording if it's.
And
Recording
we
off.
are trying to see that what.
I think you can stop sharing the screen, I believe.
Yes. 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 Just a minute.
Uh, we